Welcome everyone to the Smart Agriculture and Animals webinars. It's part of the NTU, CIA, and University of Illinois CDA, Center for Digital Ag webinar series. And this is about global perspectives on digital agriculture. Tonight, we are going to have 4 speakers, and I'll go through their names. They're all going to talk about smart agriculture and animals, starting with Dr. En-Chung, who's an associate professor in animal science at NTU. And later I'll introduce our other speakers in more detail, Dr. Angela Greenmiller, Dr. Yang Fu Kuo, and Dr. Isabella Condotta. And the intent of this is to spend about 20 minutes on a presentation, leaving a few minutes for question and answers. Please type questions into the chat if you like, or share them with the audience for the classroom that's at National Taiwan University. Thank you for joining us this evening, and let's turn it over to En-Chung.
Yeah, thank you for the introduction. Today, the topics I'm going to share with is lameness detection in gilts only, because the time limitation, so I skipped the sows part. We use kinetic and kinematic analysis together to find out the lameness or soundness in gilts. And the durability of sows would be affected by their leg soundness. Leg problems, or we call that lameness, is the second leading cause of cooling after reproductive disorders, especially during the first 3 parietes. And especially in Taiwan, the floor of pig pens, because the high temperature and the humidity in Taiwan, the pens would be cleaned by water almost every day. So the leg soundness is very critical problem for sows in Taiwan. And the observation of the structure or the prediction of the soundness of leg, during the growth process of pigs is one of necessary tasks of selection program. And majorly this done by the experienced manager on farm. So that's the lameness scores we used on the farm. The score 1 is the working normally, the pigs itself. And we can show, it's a small window here, though. Let me show that the pigs move normally. That's score 1. And once again, just every leg is evenly. Okay. And the score 2 is the stride. One of the legs affected, the stride is shortened, but it's just slightly lameness detected. So that's the second video here. Now just take a look of the right hind foot. Again, the right hind foot. Just shorten, a little bit shorten. Okay, that's score 2. And that's the score 3. The same, the hind right foot. Oops. Even we can say, we can tell the short stride and a little bit gait problem detected. And the score 4, we can see the leg affected always on the air, not touch the floor, like the 2 pictures here, this one and this one. And the score 5, the pigs already cannot move. So that's the managers detect on the farm using their eyes and their experience. Okay. And by the equipments, we have 2 different methods. One is the kinematic, using videos to detect, like the making the 3D movie. Okay, and for the kinetic, we use different equipment like the pressure mate and the force plate. And the two states we detect. One is dynamic, is moving status, and the static state is staying just on the floor. Then we detect everything we want to measure. But for the lameness, we want to detect it in dynamic state instead of the static state. So we just take a look of this part. The kinematic, we need to use this kind of mark or tap on the skin. But this way in pigs may not be very effective. We can tell from the several papers before, they found not very easy to detect the significant difference of lameness and soundness. Why? We check it by our own. Okay, this because the tap or the mark on the skin not always attached firmly to the joint point, because the skin of pigs not always attached to the joint. It's slightly sliding to the right or to the left. So it makes them incorrect measurement. And for the pressure mate, it can be more accurate to measure different kind of like the force, the ground reaction force, the stride length, the stride time, that kind of parameters. But the problem is for the practical use on farm is very expensive. I calculate a little bit that's US dollars for 60 centimeters by 60 centimeters. One set of the pressure match is $30,000 US dollar. So it's not very cost effective on farm. So we are going to make a new method to measure these kind of things, to detect the lameness and the soundness, but cost effective for the farm. So we measure in our experiment, we measure the kinetic and the kinematic and the kinetic at the same time. And using the kinetic from the video recording and measure on the video to predict the kinetic, especially the pressure mate, the GRF, the ground reaction force, from using the video. Okay. So we have the pigs. We select the sound and the healthy pigs, 12 Landrace and 12 Yorkshire, and we measure that from the 3, 4.5, 6, and 7.5 months of age. Because of the limitation of time today, I'm going to just mention the 7.5 months of age, because that's the time the farm manager select the gilts into the breeding herd. So that's the left-hand side is the pressure mate. We rented. we don't have enough money to buy it. Okay, and the two sets, because 60 plus 60 is 120 centimeters. So we can allow the pigs 2 sets continually on the pressure mate. And the camera we use just 1,000 US dollars, the ordinary camera, not 3D. And that's the setting of our walkway for recording and the pressure man. So we put the pressure man on the recording pen and the camera here, and here's the preparing pen, and we drive the pigs walking through the pressure man and at the same time recording their movement, and then move them back to the preparing pen. So we can record both sides of the legs. Okay. And that's the real situation. That's the camera here, and that's the pressure man. And we drive the pigs walking through the measurement and at the same time recording. And from the measurement, majorly we got the ground reaction force. I use GRF to shorten the words. Okay, and we can see the different colors represent the different foot. The right front foot in red, the left front foot in green, and the right hind foot in pink, and the left hind foot in blue. So we can see the software package shows all different spots on the measurement, and here is the different the GRF for different foot on different step. So that's the sequence. Okay. And here comes the peaks. That's the direction from this side. So the peaks moving, that's a little bit slow move. And that's another side, the other side, back to the preparing room. So we can tell right now, because I mentioned the tab or the mark on the skin is not very useful for pig. So we use machine, the deep learning, we cooperate with Dr. Guo. He will be the third speaker today. We cooperate with his lab, and my graduate student identify the joint on the slow movie, the video, and mark every spot of the joint for every frame. Then the deep learning model just can represent exactly the spot of joint. So using this way, instead of put the mark or the tab, refractive tab on the skin to avoid that kind of sliding problem. Sorry, oops, where is this? Okay, here. Now, that's the spot, the joint spot we want to mark. The A, B, C to F is the front foot, and the G to L is the hind foot, and represent a different joint. So that's automatically we can right now measure on the video, and especially we want the angles of each B, C, D, and E. And also for the hind foot H, I, J, K, that's with the angles on it. And these angles can be the factors of GRF. And also we measure comparing the video and the measurement. We come out the stance time and the swing time and the stride time and also the stride length. We also recording their age, days of age, and the body weight. That's for the front leg. And the angles of the front legs. And here the hind legs, the same setting. So we apply the symmetric index. The symmetric index is for the front foot, we use left foot, the stride length from the measurement, divided by the right front foot. So that will be the symmetric index for the front foot. And for the hind foot, the same thing. So we come out, and also this paper provides us the normal range for the symmetric index is 0.8 to 1.2. And the mild lameness or mild asymmetry is 0.76 to 0.8. And the other side is 1.2 to 1.24. And the severe lameness is greater than 1.25 as well as smaller than 0.75. And we use the accuracy analysis, the true positive, true negative, these kind of things. So the pressure mass tri length, we construct the index. Okay, the index shows is positive, means asymmetric. Okay, and the negative means symmetric, and we assume they are sound-led, and the asymmetric, we assume they are lameness. And the predicted, the first one is the experienced manager. He used his eyes and the experience to justify, to judge the gilts is lameness or soundness. And the second predictive is our GRIF predicted from the equations. Okay, and we also do the GRIF ratio for the front foot to identify their symmetric or asymmetric, and also for the hind foot. And the sensitivity is to find out the true positive, and the specificity is to find out the percentage of true negative. Accuracy is both. Combine them together. And here, that's the manager. By the manager, the selection results totally 11 asymmetric. He just found 4 of them. So the sensitivity is 36%. But he's really good in specificity, 100%. So total accuracy is 85%. And in our prediction equations, the ratio we use, the asymmetric, we can find 7 out of 11. But we are a little bit less than the manager is, we have specificity only 94%. The total accuracy 87%. Okay, one thing to mention, these seven symmetric, the manager cannot identify later on to the after the first parity. 3 of them fooled because of the lameness. So the conclusion we can get today is the prediction equations of GRIF using kinematic gauge analysis from this study obtained R² 0.75 for the front foot and 0.69 for the hind foot. And also, based on the symmetric index at 7.5 months of age, the sensitivity, finding out those skills with lameness from our prediction equations reached 64%, better than 36% from the experienced manager. So right now, for the further study, we are already moving on. We increase the number of gilts tested and collecting more parameters measures in kinematic gauge analysis by the video automatically to reach higher R² of the prediction equations. So that's what I have today, and that's the hard work done by Xiao Han, my PhD student, and I was the one always running after the pigs. Okay, thanks.
Thank you, Dr. Lin. We have ample time for questions. Anyone can either ask a question directly or type your question into the chat. And so I don't see, let me check the chat while we're waiting. From National Taiwan University, do you have any questions in the audience?
Probably not.
So we have 7 local questions. Anyone want to raise hand and ask Dr. Lin directly? I think we have one foreigner student here. You can ask Dr. Lin directly.
Dr. Lin, I'll ask you a question. You did your research and you had some human or managers making assessments, and then you have automated assessments, and it looked like the results were very interesting for the amount of data you were able to use. Did you get an impression from the managers of their acceptance level of using technology to do this? Is it something that they were excited about, or did they have what kind of reaction? Do they have a trust in technology, or are they skeptical?
Yeah, surely. Very nice questions. Yes, we did. And in the beginning, he just believed what he saw. But later on, after first parity, the 3 out of 7 he didn't detect with lameness, and happened to be lameness in the after the first parity, and he said, probably I need to believe that. Is that
Go ahead. I was going to say, when you think about the commercial deployment of this, if it could go and become something that they use in practice, is it something that you would try to observe pigs when they're moving from one place to another, or is it a kind of capability that you would integrate into? How would you integrate it into their environments, in your opinion?
You mean the environment to detect the video on the farm? Right.
If you did this more, if you were able to build an automated system in the future from this learning, how would you integrate it into the production facilities?
Yeah. There's a nice setting in the previous papers. I show a little bit, but I didn't mention it clearly. Yeah. Just a moment.
Okay.
Yeah, here. Yeah. They have this kind of setting. Yeah. The pigs walk through a whole an aisle, and with net or fence on the both sides, so we can record it. Just drive the pigs go through the aisle, so they will be easier. Right now, our setting just to do the experiment.
Yeah, of course. Thank you. That's interesting. We do have a question from NTU. Have you compared the reliability or accuracy of kinematic versus kinetic methods in evaluating leg soundness in pigs, especially between dynamic and static states?
Yeah. We didn't officially compare that, but in our experience and our observation, we found the measurement still reliable, more reliable than the video, because the video might be affected by the light and the shadow, and also different kind of things happen, and also the pigs direction changed or stopped, so the measurement is still more reliable, but it's very expensive.
Great. And then actually, we have a lot of questions that are being entered in the chat now, but we've used a lot of our time. I'm going to ask one more question. How can we distinguish between a pig that is truly lame and one that is just has a naturally unusual walking style?
Yeah, it's difficult.
Okay.
Especially in the stage of guilt. So that's why we put actually the original their selection program down in 7 months of age. So we postpone a little bit to 7.5 months of age to make sure they really lame or not. And the way we are right now for the final decision is after first parity. Okay.
Yeah, thank you. Thank you, Dr. Lin. And at the beginning, we had a little bit of challenges in the announcement. Dr. Lin is Associate Professor Department of Animal and Science and Technology at National Taiwan University. Thank you again for your presentation today. We're going to move to our next speaker, who is from the University of Illinois, Dr. Angela Green Miller. And her talk, Angela is a associate professor in ag and bioengineering at the University of Illinois. And her talk today is going to be on building and evaluating models for automated animal behavior observation.
All right. Can you hear me okay?
Yes.
Great. Thanks for the introduction. Again, my name is Angela Green Miller, and I'm excited about this set of talks and this potential future collaboration. So thanks so much for the invitation to be with you today. I'll start by acknowledging that this work was done under funding provided by AI Farms, which is an artificial intelligence institute here at the University of Illinois. We have multiple thrusts within that institute, and this one is under thrust number 2. And specifically, there's a fairly good team of different faculty that are working in this space, specifically on computer vision techniques to support labor needs in commercial animal production systems. And really what we're looking for is ways to give extra eyes on the animals more of the time, so that caretakers are doing their job, but the animals are also being observed to identify different types of care needs. And so some of the examples are there on the slide. I won't read all those to you for the sake of time, because I want to get into the messaging of this talk today. So one of the things that we have been working on in my lab is a lot of the methodologies for how might we utilize computer vision in order to create automated techniques to do that observation. And today I want to focus my talk on the importance of thinking about errors and how those errors carry through into the ultimate model. And so just a quick overview of what we mean when we say uncertainty of a model or propagation of errors is that we have some endpoint that we're trying to predict to get to. And the case that I'm going to go through in our examples, we're looking to ultimately identify pigs that have a care need. And so that would be our end result. But all of the inputs that go into that prediction have errors associated with them. So we might have errors associated with the sensors we're using to collect data. And specifically limitations on the precision of those sensors is one example. We might have errors that are associated with variability that comes from external factors, so things we can't control. You know, the previous speaker just mentioned things like lighting that changes throughout the day, sounds or things that might make the animals behave in a way that wasn't represented in the model we built. So there's external variability that can impact the performance of our model. Uncertainty from how we're estimating things. So for example, we might be building a model that can identify something, and there's some uncertainty associated with that piece of the model that then connects to another model that tracks something, that then connects to another model that does something else. And so each of those estimations may have an error associated with them. So we could continue for each specific case and continue to identify places that errors might come from. But essentially all of those errors factor into one sort of master error that tells you how well you're able to predict or how well your model can tell you, in our case, where the unthrifty pig is. And this really has an important implication for ultimately the implementation of models in the future. So some of the very early technology in animal systems didn't work really well. It had a large error. So it would either have false predictions, false positives that would say something was happening and then it never happened, or it would miss really important events. And so the error made it such that it didn't predict something that was really important. And so in order to build the trust and make those tools useful, we have to really think about what is the amount of error that we can tolerate for this tool to be useful. What are the sources that went into that? And then we can strategically say, can we reduce some of those sources? Can we tighten up a model? Can we put in a better sensor? And so we can make strategic choices about how to manipulate and control the amount of uncertainty going into our models. Okay, so as we're thinking about animal systems and as we think about how to build a model that can get us out to identifying a caretaker need, we have to have some sensors. And so in our case, we're looking at computer vision. So we're going to have cameras, and we could train those computer vision models to do a lot of different things. We could train it to identify animals and track those animals. We could train it to identify physical attributes. We could train it to look for biological responses. We could look at facility features or sensors that are coming from the facility. So we have a variety of places we could pull data from. But then we want to take that data and translate it into some knowledge. We might be building something like artificial intelligence models for that, things that could help us to predict or estimate characteristics of our animals, things like production characteristics, health characteristics, other welfare characteristics. And we're really, in the example I'm going to use, we're looking for challenges to homeostasis. And we'd like to identify animals that are, we're going to call them the unthrifty pig. I'm going to use pigs as my example today, the unthrifty pig. And in that case, then we'd like it to trigger some sort of an alert that you can take an action on, different types of tools or alarms or whatever to let our caretakers know there's a need. Ultimately, we want to do this to improve animal outcomes and economic outcomes. And while we're using pigs as our examples today, we would like to also have techniques that translate to other species. And we've also kind of worked on translating some of our things into smallholders with goats. I'm not going to get that far today. I'm really going to focus today on specifically biological responses of behavior. So using behavior to understand what's happening inside the animal in order to identify the unthrifty pig. So that's kind of the focus of where we're going to be in this massive space of what we could do with technology for identifying things with our animals. Okay, so we're honing it down into behavior observation. And I'm just going to show you a couple of the things that we've been doing in my lab with respect to that. How do we think about errors, and how do we think about where errors come from, and how to handle them as we're thinking about automation and automated behavior observation. So the first thing that I want to talk about is how we took a lot of, and we are taking a lot of traditional methods for how we analyze behavior and using them to inform new techniques. So specifically some of those digital techniques. The end goal, and this is my long-term dream. I don't know if I'll see it in my career, but this is the ultimate end goal. We would love to see an automated ethogram. So essentially a tool that can watch in real time many, many different things when the caretaker can't, right? So we're not going to have a caretaker there every day, all day, watching every single animal and logging every single thing they do. But if we had that level of information, we could infer a lot about animals, what's happening with them internally to identify those care needs. And there's a lot of computer vision techniques available to us that can help us work toward an automated ethogram. So things like animal identification, location and movement, which we can do with tracking, pose estimation to look at some of the physical characteristics and the postures, and all the way out to activity recognition, where we can look at sequences of postures to identify behaviors. So we have tools in our tool belt. We don't yet have models to go with all of those, but we have a lot of tools in our tool belt to work with. And ultimately, what we want to get to with this behavior observation are identification of behavior events and characteristics related to how the animals are spending their time. So what are they doing, and what they're doing tell us, right? So we can estimate parameters and movements like postures, like activities, and we'd like to translate all of that into some sort of summary. And so some of the example metrics that we might be trying to get to are things like daily time budgets. So how are they dividing up all the time in the day and using it? We can track that over time and look for deviations that might identify an emerging problem. And really one of the benefits of automated tools that we hope to capture is early identification, right? So our caretakers are fantastic. Whenever we have well-trained, experienced caretakers, they can tell us what's happening when a layperson has no idea, right? So what we'd like to do is train models that can do that. But we also want to train models that can do it even earlier than our best caretakers. Again, that's the goal. So daily time budgets, we could also look at very specific individual behaviors and look at time spent doing things. So time spent at the drinker, number of visits to the drinker, time spent at the feeder, number of visits to the feeder. Amount of time active or inactive, number of times doing things, number of times standing up or lying down, number of times having different types of social interactions, whether those are agonistic or whether they're aggressive or whether they're positive in nature or not. So essentially, we want to identify metrics that can help us better understand what's happening with the animal. Yeah, and again, we could continue that list. If we want to think of how this plays into that example of errors, we're going to step through an example of attempting to build a behavior prediction model based on XY coordinates. Okay. And so we're going to look at two components of that could be there. So our end goal is to identify the unthrifty pig. We have some amount of uncertainty associated with that prediction. Going into that model, we have things like sampling strategy. So when are we looking? Where are we looking? Are we looking for the right behavior in the right place at the right time? We might have errors in our computer vision models. For example, we might have an object detection model and it may have some error associated with it. It will have some error associated with it. We may be then taking some object detection and putting that into another model that is going to predict the behavior. So we've identified there's an animal, and now we're going to attempt to identify what the animal is doing. And again, we've got another model that has some error associated with it. And we could continue on, but for the sake of time, I'm going to stick with those three. Okay, so the first one, when should we be looking and what should we be looking for? So if we use some of the techniques that have been used in traditional behavior analyses to build an ethogram and look at sampling strategies, we can make some inferences about what a computer vision based automated tool might need to consider. All right, so the example we're going to look at is with a single pig in a very controlled setting in a research lab in the Piglet Nutrition and Cognition Lab here on our campus at the University of Illinois, and it was done in collaboration with Dr. Ryan Dilger, who works with a lot of translational models for nutrition with pigs. And that's what this facility is designed for, early life nutritional interventions. Tani, who I think I saw in the room, is the grad student who was leading this work, and she's a graduate student in animal science. She did part of this for her master's and is currently working on a PhD. Just enough context that we can move forward. This data set was on multiple 6-day-old pigs housed in individual research pens. They essentially have these little condos that you see in the picture there. They have a neighboring animal. They're divided by plexiglass, so they can't touch one another, but they can see and they can smell one another. We have the animal that we're looking for. They are fed a liquid milk diet and a milk replacer, and then they also have access to a cup waterer. So that kind of gets you oriented to their housing. Each pen contained a ceiling mounted camera. That's the view you're seeing there. And all of the pens are identical to one another with respect to where things are located. So the divider is always in the same place, and even if you turn the arrangement, the drinker is always in the bottom right if the plexiglass is at the top, like this image. All right, so the first thing that we did was to create an ethogram. I'll show you that on the next slide, but essentially that's a complete list of all of the behaviors and postures that we might observe with a definition for how they would be labeled. And then for this sampling analysis that we did first, we randomly selected 3 pigs from the whole data set. We call that our ground truth for this sampling comparison. And then using behavior labeling software, we observed and applied the ethogram to 24 hours continuously, meaning that every frame has a label of behavior or posture that is occurring in that frame. So there's a total of 72 hours of video with every frame labeled. This is the ethogram. I'm not going to spend a lot of time on it, but the first column shows inactive and active. The second column shows that there are 5 behavior and posture categories, and there are 23 individual labels that a a frame could have. So once we labeled all of the video, we look at how to then summarize it. So the summarization metrics you choose are very important to consider. In this case, we chose 3. One is number of occurrences, and it's normalized based on time. So, you know, if your video doesn't start and end at exactly the same time, we normalize for that. Average bout length is a way to measure duration. So number of occurrences is how many times did they do something. Bout length is how long did they spend doing that thing. And then the third one is percent of observations, so total percent of observations. All right, so then with this analysis, we're really interested in when should we be looking? So sampling strategies. So we have a continuously labeled data set, and we sampled from that many different ways. And so continuous sampling is where we have observations in sequence to one another. And in this case, the sequence lasted for 1, 2, 5, 10, 15, 20, or 60 minutes. Instantaneous sampling is where you look just at that frame at that moment, identify what's happening, and then skip to the next sampling cycle. And so this one is 1, 5, 10, 20, and 60 seconds. We're going to squish that down into show you 1 minute. And then we also did instantaneous sampling for the 2, 5, 10, 20, and 60 minutes like we did for continuous. All right, so those are all of our different what we called subsampling strategies, where we sampled from the ground truth. And then what we want to do is compare it back to the ground truth. Okay, so we use 2 sample t-tests with each of the summary metrics for every behavior label for each sampling strategy. Okay, so it's just a two sample t-test. We set an alpha of less than 0.05 is statistically different. If a sampling strategy is statistically different, then that sampling strategy cannot represent the full data set. Okay, so that's how we interpret those results. If there is a statistical difference, that sampling strategy does not represent the data set. All right, so here's what our results look like. So I'll orient you to this table because there's a lot in here, and this isn't all of our results. These are just some sample results for you. So the first section across the top, the label is the behavior label. So all 23 labels are there. NNO, remember, is number of normalized occurrences. So just how many times did it happen? The next label, ABL, is average bout length. So how long did it happen? TPO, total percentage of observations. Okay, if the block is colored in yellow, then it was underestimated. If the block is colored in red, then it was overestimated. And then you can see the numbers across the top are those the sampling time frame in seconds. Okay, so for many of the number of occurrences, we're underestimating how many times things happen. For some of those, for all of the sampling strategies, if it's in green, that's clear. I'm not sure what the white is actually. I could ask Sonny, but if it's green, then it is representative. If you look at bout length, lots of red, lots of not representing the duration of that behavior. And then if you look at percentages, it looks a little bit better. So sampling very quickly, and I'm going to say anything between 1 and 60 seconds is pretty quick for a traditional behavior analysis. So for someone who is manually writing down or manually collecting, even with a digital tool, but looking and observing, anything between 1 and 60 seconds is very, very quick. But for a computer vision tool, it wouldn't necessarily be that quick. It would use compute power, but it wouldn't necessarily be really quick to say you're observing once per minute. However, you can see from these results that it's important to think about which one of those metrics is important to you so that you're not overestimating or underestimating that metric. For what it's worth, for the traditional behavior analysis, we did kind of simultaneously do this exploration of automated analysis while we were doing a traditional behavior analysis. We implemented a strategy based on these results of 5 minutes of instantaneous sampling or 5 minutes of continuous sampling for every metric. It wasn't perfect, but it was one of the best representations for holistically looking at all of the observations there. We could play that forward and say a computer vision tool doesn't necessarily have to look every single second or every single frame, that maybe we could spread out those samples and every 5 minutes take an observation to reduce compute power, to reduce, the load on the system, those sort of things. And I think that's where I'm going to stop with this one, but feel free to start logging the questions on the when should you be looking and how do you interpret that. The last piece I want to talk through that we've been working on is predicting behavior from location. So we watched a lot of video, a lot of video. So we watched a lot of video on those 5-minute intervals where we have a behavior label every 5 minutes for 24 hours, and this is for 21 pigs. And so we wanted to say, can we, if we only know locations, we have a camera on the pigs, we have all these behavior labels, if we only know location, can we create any correlations between what they're doing and where they are? So again, using Tanya's data, we had another graduate student named Anu. Her background is in data science and computing. She's a graduate student as well. She first built a YOLO model to detect the pig, place a bounding box, and calculate the centroid. And so then what you're looking here is a data set with 21 pigs labeled for behavior every 5 minutes. So each one of the dots that's popping up now is the XY coordinates of that pig's location, and the color code is what they're doing in that 5-minute segment. Okay, so we wanted to know if we only have those XY coordinates, can we create a model that could predict what they're doing? Can we infer, how much can we infer from coordinates alone? All right, so if we play that out 24 hours, this is kind of what it looks like. It's a very, very messy, but kind of interesting plot. That's for one pig. We have 21 of these. So I'm going to focus more on the errors than I am on the model. So I've only picked out 2 to share with you, but kind of circling back to this idea of, the uncertainty, where is it coming from, and how do we consider that as we're evaluating those models? All right, so the first one, we did a k-means clustering analysis. The overall accuracy of the model was 82%. In that model, we put in, I think here we have 16 behavior and posture labels. We dropped some of them out because they just didn't occur in the data set enough. We used 9 of the pigs to train the model, 12 of them to evaluate so that the model could see novel data. 82%, not too bad. We see precision metrics, recall metrics, and an F1 score. Again, for sake of time, I don't want to spend much time on those, but the model doesn't look too bad. If we look at where the errors occur, where misclassifications occur, resting is where misclassifications occur. You can see kind of these hot spots, resting being misclassified for, well, the surprising ones that was misclassified for was exploring the pen and locomotion. Also, a lot of misclassifications with feeding, which kind of makes sense because they're standing still when they're feeding. Now, if we dive into that a little bit deeper, our model had 82% accuracy. We had over 170,000 samples that were used to train. However, resting is the dominant behavior of 6-day-old pigs. So more than 90% of our data were resting. So if the model had just predicted resting for everything, it would have had a 91% accuracy. And so our model prediction really isn't that good when we look at it that way. So we looked at adjusting a lot of things in our model. We changed some of our features. We changed how we combined our behaviors. We wanted to reduce the number of low representations. We went through about 50 iterations or so at this point. Our new model uses a completely different approach. It's a random forest model. It has 95% accuracy. We reduced that 16 behaviors down to, they're all still there, but some of them we grouped together. And so now we have 7 categories instead of 16. So it's looking pretty good. If we look at the errors for this one, resting is still the most. However, it's now resting is misclassified as a couple of the groupings. So groupings where they're standing still makes sense. So we have a grouping with behaviors and postures where they're standing still. It makes sense. It would misclassify with that. Feeding is still a high one in there too. And if we look, we still have the same 170,000-ish, a lot more than that, but it's 170,000 or so data points. Still more than 90% of that is resting, but our model is now better than that. So that's good. But if we dive one step further into saying, well, where are the misclassifications happening? If these red numbers that are popping up here, those are the misclassifications. So of those total samples that went into it, this is how many of them were misclassified. And if we look at the percentages of that, so for drinking behavior, 90% of the time it's misclassified. For two of our groups, it's nearly 100% misclassified. So while it's really good for resting, it's maybe okay for feeding and elimination. For the others, if we need it to predict them, it's probably not a very good model for that. So kind of wrapping up with this uncertainty piece, when we use the traditional like model building evaluation for our models look pretty good. Like a 95% model looks really good. But when we broke it down, if we needed drinking as a really important behavior, that doesn't show up as not important if we just look at the model numbers. So one of the things that we're finding, not just in these cases, but in some of our other cases as well, is that we need a more standardized approach to characterize performance and quantify errors for animal-focused prediction models. So even with the results we had, the errors of the categories where they're not resting might pass too much error into the final model to help us identify an unthrifty pig. So if drinking was really important, that's not a good model for it, but it doesn't show up if we just look at those kind of overall model prediction numbers. So where we are in this process, we're very much wishing for more established methods and would very much welcome some conversations about how do we do that, especially as we're using many different techniques and many different approaches to have a fair way to compare them and say, you know, which one really is getting us closer to this automated ethogram, which is our end goal. So with that, I will stop.
Well, thank you, Dr. Miller. There's a couple of questions from the National Taiwan University graduate students. Are there plans to integrate multimodal data, for example, audio, temperature, or RFID to improve the behavior detection?
I would love to say yes. I don't think we're that far into it. I think those are all very, very good examples. I think especially sound is a great one. We don't have a current plan to do that, but I think it's a really good consideration that we should do.
How about another question was how to collect images at night? Will the lights remain on during the night, or you try to come up with, I mean, is there light sources that the cameras can pick up that the pigs aren't sensitive?
No. We typically, they should be on a natural light cycle. Like I would never advocate for keeping lights on all the time. That you get a whole new set of problems with that. But night vision cameras work. We have a number of other data sets that we've worked with. This one too actually switches over and it uses infrared at night. So you can, depending on what the model, how it's built, it can handle nighttime as well.
I actually have a curiosity of my own, so
Yeah.
I have a question. We're doing some work where we're putting missions in a mission plan, and basically building a RAG for an LLM off of multimodal data. And I think it seems like some of the behaviors are kind of like a Markov process for a certain number of images. And I know your big picture of monitoring for 24 hours. All those dots would connect together in lines, right? And so I guess my question is there an ability to understand it's kind of like some models of behaviors that you're trying to detect, and would something like combining it with an LLM or multimodal data, would that actually provide a possibility for tying the links between the CBML sequences and the actual behavioral situation? I don't know if you know the answer, but just
I don't. And we're like always, even within our group, but with outside of our group, I love hearing other people's ideas for what would they do with that data, because it's rich data for sure. And we're trying to analyze it as simply as possible, right? So because if we ever want to get onto a farm, like right now, just the compute power is not there. So could an LLM do it? Maybe. I'd be happy to team up with somebody to do it. To see what we can do there,
It would be a similar problem on how to observe behaviors of different types of things. So yeah, well, we can talk about this.
Yeah, absolutely. I mean, it is tedious to watch video, for
Imagining.
sure.
Yeah, I was thinking that was another question I had is that so we know how to do labeling of training people for labeling, but how do you train for labeling behaviors? It seems like that's an incredible level of expertise.
It absolutely is. You know, in another study, we actually took novice observers, gave them novel data and compared it to our more trained experts. Our novice observers couldn't agree on anything they were seeing. And so we very quickly decided we can use our novice observers to identify pigs, but not to identify behaviors.
Okay, and so one more quick 20
Yeah.
seconds. What behavior patterns are the most indicative of unthrifty pigs, and what are the appropriate follow-up treatments or management strategies for such animals?
That's fantastic. We're not there yet. Man, that's where I want to be.
Okay,
That is
thank
where I
you.
want to be. Yeah. So I think in those little pigs, you know, if they're already resting 90% of the time, it's going to be really hard to detect an uptick in resting behavior. So I think actually where we're going to be looking is in some of those less common behaviors, because that's where we'll see the change and we'll be able to measure it before we can see a change in resting.
Okay, thank you for your
Yeah.
presentation, and
Yeah,
we're
my pleasure.
moving on to the next speaker. Our next speaker is Dr. Yang-Fu Kuo from National Taiwan University. Yang-Fu received his PhD from Purdue University. I forgot to mention with Dr. Lin, his was Iowa State, so we're all kind of here in the Midwest for some of our degrees. His research includes machine vision and automation in agriculture, and today his talk is going to be on applications of machine vision in animal husbandry. Dr. Kuo.
Yes, give me a minute to. Oh, okay. So how about my slides?
There you go. It's starting to show up.
Yeah,
There you go. Thank you.
Yeah. Sorry, the screen looked complicated on my side, so I don't know how to do that. Thank you very much for the introduction. So I'm Yang Fu from National Taiwan University, and today I'm going to share some experience. So the title is Applications of Machine Vision in Animal Husbandry. It's a little bit wide, big, but because I'm putting several independent topics here. So okay. So let's talk about why animal husbandry. We know that animal is actually the main protein source for human worldwide. And I'm not sure if this table is still valid since the tariff issues recently, but anyway, we will still raising animals at least for I can see that in the coming 10 or 20 years. And but animal raising is actually very laborious. We know that many farmers, they have to work for a long time in the farm, do many chores in either cow, chicken, or in pig farming. Those are the main economical animals worldwide. And so, and another issue that we face is that labor shortage. And in the lab, you can see that a chart that is the number of births in Taiwan, and it dropped from 400,000 to around like 130,000 recently. And so a drop of probably approximate maybe 60 or 70%. And on the right-hand side, we can observe in the developed countries that the employee in agriculture is dropping too. And I think this is worldwide, especially for the developed countries. And so what we can do over here is machine vision. We use machine vision
to replace what human beings need to do, and that will save the labors. And that's the like the point why I started to do this research. So I will give 3 topics. The first topic is about the monitoring of chickens in Taiwan. And we know that the chicken is a major protein source and still growing worldwide. And the chickens here I'm talking about is actually not broilers. Those are native chickens in Taiwan. And native chickens are very different from broilers. They need like 12 or 13 weeks to raise. And that's a long period of time. But the texture of the meat is very different from broilers. So for the domestic consumers, they like this kind of meat for like the chicken soup cooking, etc. And this is also true for many countries in Asia. And another issue, I think it's the same in the United States, is the avian influenza. I know there are many like egg chickens. Those are killed because the disease. And we think that this disease is going to be an issue if it outbreaks like the COVID-19. So every time that we enter the chicken farm, we have to put protections. And that is actually a lot of work and also make you feel uncomfortable. So we try to prevent the frequencies that, or the time that the farmer needs to go into the chicken farm. So some solution that we come up with is automatic monitoring and warning as also. So the basic idea is that we install some cameras in the chicken farm, and then we stream those videos to a cloud server, and we do some calculations, and I will explain the details later. And more than that, we come up with a web page or some SMS approach that can warn the farmers what is going on in the chicken farm, and only ask the farmer to go into the chicken farm when there's emergency. So what we are monitoring? We are monitoring dispersion and movement. So dispersion is actually like how the level spreading of the chickens. So in the very left-hand side, we can see there's a dispersion number of 0.4. And that means there's almost no chickens, very few chickens. And on the right-hand side, we can see a dispersion of 1.4, a high, relative high number. And that means it's crowded in that area. So this is only an area in the chicken farm. But if this area is crowded, that means in some areas there's no chickens. So that could be an issue. And another is the movement. So we use YOLO and some tracking algorithm. If I recall correctly, sort. So we know the movement of chickens. And so before we do anything, we have to model their behaviors. I mean, the dispersion and the movement using some principal models. So here we can see that the x-axis is the week. So we can see that the dispersion going up when the chicken grows. And that makes sense because when the chicken grows, they have the space between chickens is reducing. So basically, they have a narrow place to stay, so that causes the increase in dispersion. And once we know the model behavior, the long-term dispersion behavior, we can come up with a model. And here we use ARIMA. And so in the green zone, that's the 95 confidence interval zone. And in that zone, we say that it's totally okay. Farmers do not have to do anything. But there is a like red warning. That means the chickens stay there crowded for 2 hours, or there's no chicken in that area for 2 hours. Then there must be something wrong. And also, next we observe the chicken movement, and we can see that chicken movement, they drops a little bit down because it's like human beings. When we grow up, we don't move that much. We don't be that active, and also the behavior between winter and the summer is a little bit different. And so here in the chicken farm, we see one thing interesting. We see one event that the chickens running, because these are not boilers, they are native chickens, they are very active, but they are running there for 30 minutes. I mean, when I saw this video, I thought that I wouldn't run for 30 minutes nowadays at my age. So what was going on there? There must be something wrong. But we don't have enough information to know what is going on, but we know there is something wrong. So these are the chickens. Now let's move to the pig farming. And for pig farming, I believe Professor Angela Green has already mentioned about there are main three stages: farrowing, and nursery, and finishing. Okay, so let's start from the farrowing. So we want to monitor what is going on in the farrowing house. And I skipped the intro. So basically, farrowing is the stage that the piglets stay with their mother, the sow, together. Okay, so what we want to consider, the major objective is the sow as well as the piglets, how they behave, how they are doing. So our method is that because for the farrowing, it's a crate. Crate is a little space, very narrow. On the top back, we install camera, and also we stream the video back to cloud and do some calculations, and we'll see the results very soon. So first thing that we do is to understand the posture of the sow. And to do this, we have to use videos as the input for a classifier. So this is CRNN, basically a CNN cascade with a LSTM. And here we see 7 behaviors. So first is feeding, and second is standing. And these are actually very hard to be distinguished using a static image. We need videos to do that. So that this is the key. And the fourth behavior is recumbency. That is something that we don't want to see. The reason is that recumbency is that the sow hides her nipple and doesn't want to feed the piglets. And that means the sow doesn't feel very comfortable. And also some important behavior like lactating. And we separated them into right and left for some reason. Okay, so these are the behaviors that we want to observe. But what do we do? What information we provide to the farmer? We don't provide those information because those are too details, and the farmer doesn't want them to know those details. They want to have a chart that tells them everything. So the scenario runs like this. Every morning when before the farmer goes into the farm, and the farmers can have a sheet like this. And it tells them what is going on in the previous day or the previous night. And they only have to pay attention on the crate that it's some issues. So in this situation, we can see that grade 4 is very different. So the yellow part is the recumbency, and that indicates that the sow in grade 4 is feel very well. Okay, and I think this is obvious. I mean, too obvious to be observed by computer vision. The real key point is the marginal one. The marginal is the sow is doing recumbency for a long time, but not that long. But there is marginal, and maybe the farmers they themselves cannot observe the issue like grade 10. So after observing the mothers, next we observe the piglets. When we track their activities, And this is doing like group tracking. We don't do individual tracking. It's too computational expensive. And as well, what we provide to the farmer is actually the chart they can observe at a glance. So here, obviously, we see grade 4. Again, when the mother, the sow is not doing well, the piglets are not doing well. For grade 10, we can see, all right, the piglets, they rest for almost 50% of the time. That's I call marginal. This is something that the farmers may not pay so much attention, considering that the farm may have like 100 crates, like how much time he or she can spend on each crate. So this is important. Now, after I finish the farrow, let's move to the nursery. For the nursery, well, we use very similar approaches that we can see. Instead, the camera is installed on the rooftop, and also stream the video and back to the observer and to observe and quantify the numbers. So what we really care here in the nursery. So nursery is, if you recall the situation the first day, you go to school in either kindergarten or elementary school. The very first day, you see someone who is taller, bigger than you are. What do you think? You think scared, right? And that is what is happening here in the nursery. So what we concern is aggression, or say bullying. Okay, it occurs everywhere, not only in human society, but also in the piglet society. So there are some important behaviors too, like feeding and drinking. So we have a chart that shows what is happening in this crate for 30 days. And the aggression is in here in the bottom left. We can see that the aggression drops gradually, and that is a good sign. If we see something that is not what has a trend like this, we probably have to identify the pig that has some issue, I mean the bully pig, and separate it from the pen. And for the rest, like feeding, drinking, etc., they should go up, like something like this, after the piglets they adapt to the environment. And other than animals, we also do some, applications in other fields, like for tomato. Here in the left, we see a tomato application that is for tomato disease identification. And we are now working on this one to, send the output from the identification of machine vision to a RAG system with LLM to provide more information to farmers. And then in the middle, we can see some application in wood identification and with a cell phone and a magnifier. And so we take a photo of the wood, the cross-section image of wood samples, and then we can identify what it is. And on the right-hand side, we see some underwater image, and that is in a shrimp farm, and that we can identify the behavior of shrimps. So this actually the research are conducted by my student, not myself. So here are my students, and these are my the companies or the sponsors. And what I actually do in the lab is not doing research, but I brew coffees. So if you need a cup of coffee, please come to Taiwan and stop by my lab. And of course, I can treat you a good cup of latte. And this is my research. Thank you very much.
Thank you. Are there any questions? I don't see anything in the chat yet. Anyone would like to ask a question online? I have one back in the examples of, especially in the nursery, looking at multiple pigs seems a lot like almost like agent-based modeling. Have you ever tried to apply that to understand the differences in behaviors, or they look like a lot of individual agents that maybe some of these modeling techniques might explain why they're going to water or playing or different things like that. I don't know if that's a tool that can be used or not. It just occurred to me.
Well, actually, certainly that's one direction that we want to do in the future work. And now for the nursery, it's not complete yet. We are still working on this one. There are more behaviors. First, we have to identify like playing. Like piglets, usually they play either with each other, and that is more challenging because we have to identify if it's aggression or play, or they play use some toys like for the piglets. And so that's what we are doing right now. But for the next stage is probably something as Dr. Reed just mentioned, some agent model or some I would say it's a time sequential model, because now I'm not doing time sequential. It's more computational expensive. But I think that's something that we can do in the near future if we have enough computational power. Yeah.
So we have a question in the chat from National Taiwan University. What are the feasibility and limitations of implementing an automated detection system in commercial farms?
Well, that's a good one. Actually, I think the farrowing project is more, I would say, complete. Let me go back here. So, what I'm thinking here is actually a total solution monitoring system in farrowing house, and the farmers can install a camera on a crate actually, and we can do some say not edge solution. I would say it's a fog solution. We stream all the cameras, their videos into one powerful computer with GPU in the farm, and then every day or every few hours, the computer gives the results of the observation on each crate for the past few hours. And now we are doing only the after farrowing or after delivery, because the sow and piglets are there already. But we can do like during the delivery or before the delivery, like to estimate. So it's not complete, but this can be something that be a solution for farmers to, so intelligent, so smart, that tells them what is going on or what is going to be happen next, and that will save their their energy to they can concentrate their mind on something else. That's something that I can come up with.
Thank you. Is it possible to fuse additional sensors into tracking, such as I think they mentioned in the chat an IMU? So I'm thinking they must mean by putting other kinds of sensors on the pig.
Yeah, I think it is possible. It is just that doing like multiple sensors, or called sensor fusion, is actually even more complicated. The graduate student have to match them one by one. So I'm trying to find a solution, a balance between what we can do and also the workload of the students. But I believe that certainly that's something that we can do. There are many people that use these IMU sensors to do like individual behavior analysis, and mostly it's for like cow or adult pig, because they are bigger and they are more mature, and we know they are more expensive, and we need to know their like individual behaviors. But machine vision, I know, well, maybe, I'm not sure. Yeah, I think that's a solution, but I'm still thinking.
And in the case of piglet behavior analysis, how do you handle the occlusion or overlapping of animals in the video feed?
Yeah, occlusion occurs a lot. If we take the look, see the rail, say for the piglets, we can see rails over there. But as long as the video is clear enough, the issue there is that we have to wipe the camera, the lens, once in a while, say at least once per week. And they still need some labor, still need the farmers to help because it's underdeveloped now. But for now, for rail, like this thick of rail, and we are doing fine. We are doing fine. So occlusion mostly happens is that that heater over there in the bottom left corner. There's a heater lamp.
Seems like it does a pretty good job with occlusion, and you can see in the images on the left. It's handling them from a top view at least, right?
Right. The view is actually very important. The view. So we spend a lot of time like choosing the optimal view. Yeah.
Okay, we have two more questions. If an abnormality is detected, are there any measures to deal with it? So you're creating chart. Well, let's just leave it at that. You have an abnormality. Things that aren't showing up in, I guess it's maybe possibility it's not in your classes of things you're looking for.
Yeah, that's a very good question. Actually, now this result is only on 10 crates. Now we are collecting results from hundreds of crates. And that is the next step. Like for the chicken application, I use like 95 confidence interval for like normal. And beyond those 95% is abnormal, so abnormality. I think from my definition, that could be something we use big data. We have enough data, and then we mine from the data to understand what is going on there. And we are still collecting a huge amount of data, and I believe we are heading to that direction too.
If on this chart here, if farmers see this, I mean, farmer has 5 or 10 crates and they come in the morning and see this, which of these charts cause them to be concerned? Or, how do they interpret where there could be an issue? Is there one of these crates that it seems more problematic than the others, or is it just different behaviors in different crates?
Yes, like the yellow part is recumbency, so we don't want the piglet to do recumbency a lot. Right. So that's one issue. And as the previous question, so if we see something like this, now we only provide a shot, but if we see something like this, we can actually provide a text and telling them, okay, now this is a warning. This is some abnormality behavior that you have put to pay more attention. And that can also connect to the RAG system that we have been talking about, that provide more information, some abilities. And if we have sensors like humidity, temperature sensors in the field, that we can even provide more precise information. There's a lot of things that we can do.
Okay, and last question. For multiple object tracking, what has been done to prevent ID switching?
That is too professional. I think the students are talking about this. So ID switching is actually occurred by the occlusion. Basically, when the piglet they go through the rail from one side to another, and like the computer vision algorithm may not know this is the same piglet. So that's ID switching. And currently, we are not dealing with that because we are calculating the total movement. In a sense, we only want to know like how active the piglets are by like calculating the distance they have moved in the past few hours.
Yeah.
But we want to do individual piglet tracking. That is Thomas Norton, professor in Leuven, Belgium, has been talking about that we have to deal with like the ID switch very carefully. So it depends on the object that we want to do. And that is something in my mind, but now I don't have a solution yet.
Thank you very much, Dr. Coral. We'll now
Okay.
move on to our next speaker. Our next speaker is Dr. Isabella Condotta. She is going to talk today on precision management of animals, definitions, challenges, and opportunities. Isabella is a professor, assistant professor in the Department of Animal Sciences at the University of Illinois, and she has 14 years of research experience, more than 14 years, focusing on animal stress, improving animal well-being, environmental sustainability, and helping farmers make better, more efficient management decisions through precision animal management. Isabella, let's turn it over to you.
Screen. Can you hear me okay?
Yes, we can.
Can you see my full screen here for presentation?
Yes, we can.
Okay, perfect. So I'll start. This is, I feel like, more of a overview of what precision management is, and I will show a few of the applications we are developing here within my group. And some of those you will realize they overlap a little bit with the previous presentations. So hopefully you're still hanging there for the last presentation. And I can show our results here or our applications here with some computer vision as well. Start with the problem that we're trying to solve, backtrack a little bit. So we all know that our world population is projected to increase almost 10 billion people by 2050. With that, we do have an increase in animal products consumption. And what that means for us on the animal science / technology realm is that we need to help to increase efficiency of our animal production systems to feed that demand and to address the increase in demand. But we need to increase that efficiency while still maintaining animal health, improving animal welfare, improving the environmental sustainability, helping farmers to take care of their animals. Generally speaking, farms are becoming larger in animals that caretakers need to look for. But still, at the end of the day, keeping the system profitable. And that's when I start talking about implementing technology within our production systems. And we can call that precision management of animals or precision livestock farming. And the formal definition of that field is the management of individual animals by continuous automated and real-time monitoring of the health, welfare, production, reproduction, environmental impact. So keyword there is really real-time or near real-time. And we really want to continuously monitor individuals so we can actually make better decisions. So how the systems work. This is a general overview of a system. We have a process, which is our animal. We generally have information about the microenvironment in which this process or this animal is inserted on. And we have information about those animals as well. Generally speaking, we might have health information, breed, sex, age, etc., etc. And with the help of sensors, we can monitor their dynamic response to the environment that they inserted. With that, we can get data to input into algorithms that will output maybe a management decision or some sort of alarm or just health reports on those animals. And this output is going to be then sent to a controller. This controller can either be automated or manual. And based on a desired output, meaning maybe how much an animal is gaining of weight, maybe how they are behaving, we can modify our environment. And with these modifications, we get a new response from those animals. And then this generates a feedback to the controller, and we can iterate this process over and over. Now, let's talk a little bit about developing tools for this kind of system. First of all, you cannot control what you can't measure. So there's a challenge there, which is monitoring the animal's physical condition in a fast, efficient, continuous, and non-invasive way. The non-invasive and the continuous there are very important. And obviously, we want to be efficient and fast, especially if we have several animals to monitor. A solution would be then to transform our most commonly subjective methods of measurements into more objective methods to gather more objective variables. Lots of our production systems use more visual assessment of animals. So we want to provide something more reliable that's not as dependent on view of our caretakers. We can do that, this assessment, through the use of sensors. Here are just a few sensors that we could potentially use. I'm using a dairy cow as an example, but obviously some of the sensors make sense for other species and some of them will not make sense. Dairy cows are just larger animals, stay longer in the production system. So generally speaking, there's more wearable sensors available, such as ear tags, collars, tail rings, nose bands, electronic boluses, and leg straps. And those generally will have maybe a combination of an accelerometer or a temperature sensor that's going to be measuring different things such as activity, positioning, maybe rumination time if it's a ruminant animal, etc. And they provide good information for us, but oftentimes this is not practical from a commercial production standpoint to have all animals having wearables or even more than one wearable. That's not really what we want. So there's another kind of sensors, which are non-wearables that include mostly cameras and microphones. So we get image and video data and sound data that we can then analyze. In my lab, we've been doing lots of computer vision research applied to livestock systems. Let's just take a look on a few computer vision applications, and then I'll show a few examples of what we've been doing within each of those applications. So image classification, we have an image and we have a label attached to that image. So for example, here we have a cat and then label cat, that's image classification. The entire image receives the label. Then for object detection, we are going to have that same class, but then a location attached to it. And that's when we start seeing those bounding boxes or those rectangles around our objects, and we can retrieve that spatial information as well. It's very useful for a few applications, but oftentimes we need to actually do segmentation in which we understand not only where the animal is in general, but also the animal edges, or maybe not even only the animal, but specific objects within our production environment. And that's when we start talking about segmentation, which can be divided into semantic segmentation and instance segmentation. Often we are going to use semantic segmentation for animal applications, which will define each individual item from a specific class as being different from one another. Now, the last application is key point detection models. In our lab, we've been using this type of model for detecting joints of animals for example, gait analysis. We also have a project going on in detecting specific locations of the heat structure of cattle to correlate that with body condition score. So I'll show a few applications now that we've been doing in our lab. Some of these solutions are either already published, a few of them are submitted for publication. Some of them will be presented in the upcoming conference in the US, the Precision Livestock Farming Conference in Nebraska this year, later next month. This is an application of detection. This was a master's student project. The first thing we wanted to know is if we could reliably detect our cows. And we set regions of interest within this pen to determine first where the animals were spending their time. So we have a drinker on the back of this pen. We also have beds over here, and we had another camera checking for the animal's feeder, and I'll show the other view in just a second. But pretty much, we wanted to develop a detection model to find cows, and this model was a YOLO architecture model and performed very well, mean average precision more than 0.99, as expected, because it's not a very challenging environment. And cows in general, they don't pose as much occlusion issues. This is a continuation of that project on that same pen towards the front, where you couldn't really see with that camera. We have a top view image of feeders. Those animals have individual feeding bunks. And we were measuring time that the animal spent at the feeder, how many visits of the animals to the feeder. So just a detection algorithm detecting the cow, and we knew based on which feed bunk it was, we knew animal ID based on that, because only one animal could access each feed bunk. We also did an evaluation of the feeding, the feed itself for volume and some other features that were extracted. And with that, we were working on predicting dry matter intake of those animals. This is just an example of the detection. And then we did some behavioral time budgets for the different types of behavior we're analyzing. Another project we have here going on is feeding behavior, but not for sows, very similar to the previous one. Let me see if I can play this video. So here we are detecting if the animal was at the feeder. Classifying an animal as feeding, it's quite complicated, but we did label a significant amount of images to really understand when the animal was actually feeding, meaning with the head inside the bunk and actually eating, and then when it was not. But it's very complicated, especially this type of view, because you don't necessarily know. Let me see if we have here, for example, at feed or not feeding, then the animal was just standing. So we had a total of 6 different postures / behaviors that we were looking for that sow, specifically in the fairing crate scenario. Moving forward, we have within that same type of data set, the video data set that we have, we wanted to detect sow nursing behavior. So in this case, we are combining postures and behaviors. If I remember correctly, we had a total of a combination of nursing and not nursing, and which are 2 different behaviors combined with the 6 different postures. So for example, here the sow is standing, which is the posture, and not nursing. A little bit further, we're going to see the sow is lying on the right and nursing. And I think you saw lying sternally as well and nursing. So we had a combination of the behavior of nursing, not nursing, with different postures of the animal. This was also a YOLO model. I believe this was a version 8 of the YOLO architecture. Now, segmentation applications. Here's an example of using segmentation for pigs. I think you can see on the first image, this is a depth image, so it gives us volumes. And we have the segmentation of the body of the animal. And then without the head, we have this shape from the body of the animal. And we use that to determine some curvatures at the back of the animal. See on this little animation, some of those curvatures. And also after segmentation, we were extracting body dimensions other than just the curvature. This body dimensions such as different heights, because it's a depth camera, we can acquire heights and then lengths and widths at the animal body. And correlating that with body condition score, We also correlated with back fat, but I did not bring those results here. So we were our mean average precision. It looks okay-ish, but considering that this was our standard was a caliper that placed body condition in a scale of 1 to 20, being at an average of 1 unit, it's not too bad considering that most visual scales are actually from 1 to 5. So this is just a bit more detailed versus a visual assessment. Here, example of using body dimensions for weight prediction as well, same type of analysis we did before, and then we were predicting body weight. So this is a top view image of animals coming to a drinker. So we have a total of 4 drinkers over here. This is just a color image, and I'll play the video for you to see animal segmentation of the body and then prediction of weight here in kilos on the left. And then we had an RFID system going on to detect which animal is which. Another example is movement patterns. Obviously, there was a presentation earlier on lameness. This is something that we explored as well from top view images in this case. And we were looking for specifically the movement of the head and then left and right side of the shoulders and the hips. And you can see here, for example, as the animal walks, we can determine these different patterns. And we could correlate this movement patterns with lameness, just 3 levels of lameness. We didn't really have a lot of animals that were lame, so that was the setback from this study. But we could determine if animal was either severely lame, moderately lame, or not lame with about 87% accuracy. Another example of segmentation model we are using to detect piglets and sows. And the goal here is to predict overall activity level of those piglets. This is part of a major project that we are working on, determining mother ability. And one of the variables that we are studying is piglet activity, along with that nursing behavior of the sows that I showed before, along with the feeding behavior of the sows as well. We have several different behaviors and postures we are studying over time and creating different time budgets to determine a sow's mother ability. Another type of posture and behavior, piglets at nursery stage. We are detecting feeding and drinking behavior along with some posture, just like standing or lying down. And we have good correlation between amount of time spent at feeder with the animal performance. We are monitoring weight of those animals, and we are getting about over 90% correlation between the time spent at the feeder with the growth of the animals. So that those are promising results there. Key point detection models now. This is just an example. This was a previous video, so it's not the best video that we have, or I should say the best model we have, but it's just an example of application. We are trying to find these key points on the animal body for gait, but not necessarily for lameness. We are actually looking for correlating the gait patterns with body conformation of these animals, and then to predict later if they are going to develop lameness or not, but also to guide genetic selection. This is a work in partnership with Dr. Josh McCann here from the University of Illinois. Another thing we're doing is checking those variables over time. So it's very important to understand that individual level observation. It's of more interest on this type of precision system than just averages. This is just an example that I like to bring of pig's mass over time for different genetics. So it's important to understand how the average animal should be growing. But also if we start looking at individual level, here is body weight just analyzed every week for an animal of this 2017 genetics here. You can see that it has a similar pattern, but obviously it's not perfect, not the same. Each individual will be a slightly different. That's why average is an average. And especially what we're looking for is potentially analyzing those patterns over time on a daily basis, possibly, and understand and use that information. Let's say that in this point where we have a slight dip on body mass, maybe if the animal continues losing weight, we can then take an action earlier than we could potentially if the animal was visually losing weight. Maybe at that point would be too late to do something. Some challenges now to finish this presentation. Animals are complex, they're individually different, they're time varying and dynamic. That means that we have to consider most, in general, animal personality and animal behavior when we are developing this type of system. For example, here we have an expectation of an animal image, a pig image, and we develop a system that will always analyze pigs in this position, but in reality, pigs can actually act like this. This is a pig jumping, so we don't have a full animal over there. And then our model, prediction model, if we are not taking consideration this type of behavior, we might predict, in this case, body weight in a wrong way. The environment for pig farms and general farms is always challenging for computer vision applications, especially. You can see pigs all pink. If we are using color-based images, you will see that this color of these pigs half gray, half pink. Here we have dirt on the pig as well. So we had to keep in mind this type of thing, especially if we're using color-based system, which we're trying to avoid. Environment as well, trying to bring indoor applications to outdoor applications. This is an example just of application of doing computer vision outdoors. That's probably not the best application that's out there for this kind of system, but there are some advantages, especially if you can put stationary cameras outside. Collaboration is always both a challenge and an opportunity. It's a very multidisciplinary team. Here we work in teams of different people from different backgrounds, and I think that's how research in this field should go. And almost to finish, measure what's important. Do not measure what, don't make important what you can't measure. That means that we are sometimes trying to measure things that are not necessarily our final destination. And I'm going to showcase this in one slide. So we want to gather data and measure that data. But at the end of the day, we want information. We want decisions. This is just an example of one image that can provide almost 400,000 numbers. If we collect those images in a 30 images per second rate, we have 11 million numbers per second being generated. And on 24 hours, we are reaching the trillion numbers. And when in reality, maybe we just wanted one number animal weight. So this real-time assessment and transformation of data into information is very important. And also, we need to be led by customers' needs in the end of the day. So you need to keep in mind if the systems are going to be adopted. End of the day, this is what we want. Data integration. So animal data should be processed and transformed into information. And this information should be transformed into decisions. And these decisions should be transformed into actions. And then there's some outcomes that we're looking for, which are more efficiency, less working time, better work-life balance for workers as well. And to summarize, precision management of animal systems, they are helping farmers. They should provide help for farmers, not really replace farmers. They should provide real-time monitoring. Privacy concerns exist, so we should be taking consideration that real-time analysis of our data, not really saving data for postponing the analysis. Farm environment is challenging for this kind of application, and we need collaboration in this field. Just a thank you to my students. I probably did not mention all their names, but they are the ones doing some of most of this research, and just some funding agencies as well. Thank you.
Thank you. There's a couple of questions already in the chat. Could this individual tracking be integrated with AI-based detection systems to predict health or welfare issues early?
I don't see this question here. Could you just repeat, please, John?
Okay, yes. Could individual animal tracking be integrated with AI-based detection systems to predict health or welfare issues early?
Yeah, so if you can track the animals, that's a better step than just detecting. So we do have a few the video that I showed from the nursery pigs that we were detecting at the feeder or at the drinker. We were tracking those animals. It is a smaller group, so it's easier to track those animals. We didn't really have a D switch at all. We had groups of 4 and 6 animals. If we can do that type of tracking for longer periods of time in pens with more animals, then that would be ideal, because then we would be not only determining what the animals are doing and having time budgets for that, but also we have an activity level, and it's a deeper information that we have. So that would be, yes, very useful. I don't think we are quite there yet with our tracking algorithms. We're getting close, but not quite there yet.
Super, thank you. Second question is, this is actually, I may re-ask the question, but it's about a YOLO model predicting precisely the identifying the position of an animal, standing or lying, in an overhead view. But of course, from projection theorem, the legs are on the other side of the animal, so you can't see as much information about the legs. Can you talk about the consequences of different viewpoints and the trade-offs in terms of whether you want to view overhead versus sideways, like for the gait motion?
Oh yeah, so I mean, for the pig systems especially, we try to put the cameras as far from the animals as possible to begin with, and depending on the farm, you might have occlusion not from animals, but occlusion from different items within the system. So when I can, I just put cameras above the pens to have a full view, but not everything is going to be analyzed from a top view image. There's some benefit of having maybe a side view as well. Complete side view, I think it's very hard to implement on pens, especially fairing crates. You can't really have a complete side view of an animal like that. You will have to just put an overhead camera. But the trade-off there, I would say, is more related to some specific activities. So, for example, on that feeding behavior, there is occlusion from the animal on top of the head of the animal is in front of the feeder. So you can't really see if the animal is chewing or not. It's just an estimation, right? So there are trade-offs. I feel like the thing to keep in mind is that we need to make our models as generalizable as possible, and consider that the camera is not always going to be positioned in the perfect way. So we either need to put a constraint on that model and say this needs to be a top view camera, or this needs to be an inclined view camera or a side view camera, or understand that we will need to feed our model more images and more examples of images from different perspectives. Oftentimes using multiple cameras and stitching those images together could provide more granularity on this data set. But obviously there are some things that we cannot see from a top view, and there are some things that we cannot see from a side view. So more the merrier in terms of view, but we need to take in consideration that each farm is going to be a little different in layout, and we might have occlusion from equipment, not only from animals.
Thank you. I think there's one more question. Will the light source facing the camera affect the detection accuracy? So we normally think about reflectance, but I think in some cases in these environments you're looking directly at a light in the room or something like that.
Yeah, I wish I had an example here. So yes, the
actually slide 17 is what they reference, so maybe they saw something in that slide.
Okay, maybe I can go quickly back to slide 17. Probably easier to just escape from.
Oh, there you go. Yeah, okay. Yeah, you have some lights in the room.
Yeah, so the short answer is yes, we have issues with light because obviously if there's no light, then you don't have a color image. But we do make sure to include different light conditions on this model, including infrared streams, not only the color streams. So I don't know if I have a good video here. Maybe I have on this other presentation if I pull quickly. But oh yeah, here, maybe I should. Can you maybe see this screen here?
Yeah.
Okay, so this video, can you see that's a bit darker? This is night and there's illumination over here. So there are cases in which it gets completely dark and we have to use the infrared stream from the surveillance camera or the depth camera. And there's cases in which we have still when the farm, it's not a farm, we still have some sort of light source that does not necessarily switches to the infrared of the camera. So we do make sure in our data sets when we are labeling this data to include all of the variation. And if it's not possible to collect this data, we use some augmentation as well to simulate different light condition.
Okay, and we've reached the end of our time. And although Dr. Casey Ting asked probably the most important question is, what are the most important actionable insights provided by the automatic monitoring systems? Will there be automatic systems to take the management actions? And I'll leave it to the speakers and other students to share those information through direct contact after this meeting. I want to share my screen one more time, if I can, and let's see if it's sharing. Are you seeing my screen?
Yes.
Great. I'd like to thank everybody again for joining this webinar. It's the third on smart agriculture and animals. This is a joint seminar webinar series for this year through the Center for Intelligent Agricultural Education and Research, the CIA at National Taiwan University, and the Center for Digital Agriculture, CDA, at the University of Illinois. Our last event will be a webinar on April 30th in the US, May 1st in Taiwan on smart agriculture and crops. And if you go to the CDA website, Center for Digital Agriculture, UIUC, you will be able to see recordings of this event and all past webinars. Thank you for joining us tonight. Thanks, everyone.
Thank you all. Have a good night.
Night. Good day.
Day.